Science is at the heart of drug discovery. Atoms and molecules are at the heart of what is a drug. A drug is a molecule with therapeutic interest, and the molecule is a bunch of atoms, the exact sequence of atom that binds to the right protein or therapeutic target responsible for the disease and blocks the disease. So, at the heart of drug discovery is finding the right molecule that binds to the right therapeutic target and blocks the disease, so to cure. And this is done with a lot of atomic interactions and, at the end of the day, mathematics and quantum physics.
All right. So hello, Maximilien, and welcome to Impulse. I'm glad we're finally able to record the episode. We missed each other in Paris when I was attending HIMSS Europe, and we initially wanted, to have the conversation face to face in your premises. So, we'll have to make do with the online recording setup. But I'm sure, will still give us the opportunity to have a very interesting talk about your path and, the work you do with Aqemia. I don't want to spoil, our listeners too much, but, for context, I'll mention that, we've had so far 2 guests on the show, with whom we discussed the use of AI for drug discovery. The most recent one being Jean-Philippe Vert, who you probably know from Aqemia and Bioptimus. He's also in Paris and London, actually, I guess. So it was also like, an eye-opening conversation, on how they are using AI to identify new drug candidates, new diagnostic markers, and also, build what is one of the first foundational models to understand biology in a new way. The second, episode was with someone called Matty Gill from Ion Labs. I don't know if you know him as well. But there, it's a venture builder. So we discuss, how they identify and, support emerging companies in the field and, the potential that AI has for drug discovery in general. The one element, that caught my attention, when I was preparing the episode was, when I was reading about your activities, and that we did not talk about into these episodes was, quantum physics. So it seems like, you guys are trying to, combine quantum physics with generative AI to approach, drug discovery from a different angle. And, although I know very little about quantum physics, I'm looking forward to, hearing from you about the topic in general. So you need to educate me and how it is, also serving the purpose of, developing drugs quicker in the future. But before, I, ask you how all these things, relate to each other. And, for us to get to know more about yourself, I'll start with an invitation for you, Maximilien, to present yourself.
So, hi, Mathieu. My name is Maximilien Levesque, and I'm the co-founder and the CEO of Aqemia. And we are a Paris and London-based drug discovery TechBio. And our mission is to invent preclinical candidates leveraging what you said, physics, statistical physics, and quantum physics.
Awesome. So maybe you can tell us a bit more about where you come from, because I think you have a previous career in research where you worked exactly on this topic. So it would be good for us to understand a bit, what was your research focus and how that, evolved into what Aqemia is doing today.
Yes, at the origin of Aqemia is a technology that was born in fundamental research in stat and quantum mechs, so statistical and quantum physics. Before Aqemia, which is 6 years old, I was a professor at École Normale Supérieure and CNRS in Paris, driving researchers around what happens at the atomic scales in so-called condensed matters, which is, in fact, what happens in liquids.
Okay.
I've had a career there. And then before that, I was working at the universities of Oxford and Cambridge in the UK. And before that, I have a PhD in theoretical quantum physics from the Commissariat à l'énergie atomique, which is the French Nuclear and Atomic Research Center.
Interesting. And so the link between quantum physics and its applications in healthcare, was that something that also came up in the research you were doing?
In fact, not at all. The funny thing, I was doing quite theoretical and mathematical applications around the physics of liquids. I solved a very old, in fact, equation nobody had solved before, called the molecular Einstein-Zernicke MOZ equation.
Okay.
Got a prize from the American. So I was applying it to the nuclear industry because it was my background. And I got a prize for this mathematics, those new mathematics from the American Institute of Physics. This prize triggered the interest of two leaders in drug discovery, in software development for drug discovery. So they came to my lab, proposed to buy the technology. I said, but why?
Mm-hmm.
Why is this technology? You don't do nuclear applications.
Yeah.
And they were eyes openers. Do we say that in English?
Yeah.
So
Eye openers.
they triggered. They taught me that those mathematics could drastically change the way we invent and find new drugs. And so it was for me the beginning of a new quest, which can be summarized into being useful to something in the universe, which translates into applicating my equations to finding new drugs and to cure patients at the end.
And so, that kind of like takes me to the initial question that I wanted to ask, what is actually, the link between quantum physics and, these maths that you were, diving into during your research and, the applications that, these people saw in, in your work and its applications for drug discovery?
So there's a direct link. Science is at the heart of drug discovery. Atoms and molecules are at the heart of what is a drug. A drug is a molecule with therapeutic interest, and a molecule is a bunch of atoms. The exact sequence of atom that binds to the right protein or therapeutic target responsible for the disease and blocks the disease. So, at the heart of drug discovery is finding the right molecule that binds to the right therapeutic target and blocks the disease, so to cure. And this is done with a lot of atomic interactions and at the end of the day, mathematics and quantum physics.
Interesting. And so your idea is really to, I'm trying to understand a bit, along the process of drug discovery, which, I think we know is like extremely complex. You have to identify, the therapeutic target, you have to identify the active ingredient that would match, and then you have to do all these, clinical studies. Where, do you see the potential of, the technology? I mean, first, I think we need to talk about the technology and how it works, and then how it, serves certain aspects of the drug discovery process.
Mm-hmm. So at the heart of, a drug is literally a molecule that binds to its target and doesn't bind to
the rest.
The rest. Binds to its target means it blocks the disease. Binds to the rest means it is toxic. At the heart of, binding is measured with something called, it's a beautiful word. It's called the binding free energy. And literally, we are the best in the world to predicting binding free energies because we are the only ones having those mathematics. Those mathematics are a key ingredient to predicting if a molecule has a good binding free energy. So does it bind or not to a target or to the rest? Mm-hmm. So in fact, to summarize the technology at the heart of Aqemia, we have a generative AI that invents new molecules, many new molecules. And those molecules are proposed to those algorithms of physics-based, quantum-based algorithms that test each of those molecules atom after atom. The physics gives the feedback to the generative AI, which learns and invents better molecules. And there is this loop of continuous improvement, invents, tests at the atomic scale, gives feedback, invents better molecule, tests, gives feedback. And at the end of the day, when we have invented and tested in the computer, we test the molecules in the wet lab and fuel another loop. And that's what we do. This way, for instance, to make things clear, we identify a new target responsible for a cancer, say neck cancer. We have a program at Aqemia. We have a program in oncology against neck cancer. This cancer is triggered by a therapeutic target, say a kinase, for instance, a given kinase. This is the only context we give to our technology. A GenAI invents molecules that should bind to this kinase and not the others. The physics, the quantum and stat mechs, they test the molecule in the computer, gives feedback, and then when we are happy from what is generated by the GenAI, we test in the wet lab.
Interesting. And so it always starts with, let's say, a therapeutic target for which you look for the molecule that will bind to this, having the lowest binding energy.
So you're very good in stat mech.
I'm not so good. I got off the term.
But minimize the binding free energy. You didn't tell me that, Matt.
Some remainings from my engineering studies, maybe.
So sorry, what was your question?
Yeah, my question was, do you always start the process from, having a therapeutic target in mind for which you design a molecule that will bind to it?
Yes, exactly. So in fact, the very beginning of the process is a disease. Then from this disease, there is known, we do not invent biology at Aqemia. So there is a known problem, the pathways, the biological pathways, the biological mechanisms are known, some is known, and some targets, some proteins along the pathway are identified by Aqemia or by partners as the reason as inducing the disease. This is the starting point of a program at Aqemia, the therapeutic target. In fact, what we do in an expert vocabulary would be we do structure-based, very rational drug design.
Makes sense. And my follow-up question would be on this transition stage where you say you've done, enough iterations to have a drug candidate made by, artificial intelligence that could be relevant. How is this then translated into a physical, compound that you can actually test in preclinical models and in the future potentially in clinical models?
So you are very right. What we do is there are 3 steps. We invent in the computer, we test in the computer, and when we validate in vivo or in vitro, which means in the wet lab. So your question is about how we move from the to the wet lab.
Exactly.
In fact, the kind of molecules that we invent at Aqemia are so-called small molecules, so chemical matter. We do not have internally at Aqemia the resources to synthesize those molecules, but we have partners all around the world, in the US, in the UK, in Europe, and in Asia, to synthesize and test the molecules in vivo or in vitro.
Makes sense.
I didn't say it, but we are a team of 70 people today.
Yeah.
60 in Paris and 10 in London, going fast in both countries. And everybody is linked to designing the compounds one way or another. Is it from the business side to the physics or quantum mathematics side, etc., etc. But nobody's in the wet lab.
Interesting. And this synthesis stage, does it always land with the exact same compound that was designed in the previous stages in terms of the atomic structure and its components?
The short answer is yes. In fact, the process, so the molecules that are generated by the artificial intelligence are usually, we say, chemically feasible. Sound, an expert can build them, can synthesize them in the wet lab. It's really most of the time the case. We also have internally medicinal chemists that were at the wet lab and that help us design the right generative algorithms so that every single molecule we design can be made into the wet lab. And then at the end of the day, we have partners, for instance, in Europe or in the US, that synthesize the molecules. And those partners, they are real partners. Of course, they work for Aqemia, but they also give us feedback on the feasibility of the molecules. And it happens sometimes that some molecule may not be doable. And that's absolutely not a problem because the process is very efficient in designing new molecules.
And I assume, you mentioned at the beginning, your work is to find or create those molecules that not only bind to the therapeutic target, but also not bind to all the rest. Is this like, let's say, selectivity? Is this also something that's part of this first step of the process, or it comes down the line? Okay.
Yes, it's in fact, I think, I like to think we are the best in the world to build high binders, to invent molecules that are highly binding and highly selective, highly effective and highly selective. And I think this will be, I will give you an example. We were talking about this kinase earlier.
Yeah.
Some kinase families are, so members of the families, take the CDKs, CDK1, 2, 3, 4, etc., etc. They are very similar to each other. And you want to bind to only CDK2, for instance. The difference between CDK2 and CDK1 is very, very subtle. It's just one or a few atoms in the binding pocket of this huge protein. The fact that we work atom by atom at Aqemia and not with an AI black box, atom by atom, makes us the best. I like to think we are the best in making very selective, for instance, CDK2 molecules against CDK1.
Okay. And so you mentioned that this was part of a program for neck cancer, right? I was wondering, how do you choose the indications where you want to, leverage your technology? Are you open to any indication, virtually, or?
Yes. So first, I didn't say that the CDK2 programs we may have or not is linked to neck cancer.
Sorry. I made the link. Sorry.
CDK2 is linked to breast cancer, especially. So
okay.
It was just an example, maybe not a good one to illustrate the selectivity issue. To come back to your question. So sorry, what was your question?
The question was, how do you choose the indication areas exactly?
Yes. So the technology is agnostic of the therapeutic area.
Exactly.
Agnostic of the protein family, because for our technology, those are atoms connected to each other. So, nevertheless, Aqemia today is mostly an oncology company with extensions that start to pop up in central nervous system disease, so
neurology,
and metabolism. And the reason why is very simple. We have had, and we continue to have partnerships with pharma companies. For instance, for what is public, we work with J&J, Servier, Sanofi. We have an ongoing large collaboration with Sanofi with a deal up to 150 million with them for a given number of research collaborations. And what happened is the first collaboration programs we've had were in oncology.
Yeah.
And so we developed internally a know-how in oncology, and we continue to push this know-how. So today, the internal programs of Aqemia are mostly oncology, and some of them are in CNS and some of them in metabolisms, but mainly oncology, even if the technology is agnostic from the therapeutic area.
Super interesting. And so in which phases currently of the drug discovery process are some of these drug candidates that are the most advanced in your, pipeline and in your, as part of your own, therapeutic programs?
Yeah, so for the fully internal program, fully wholly owned programs, we have a funnel of preclinical programs. The three most advanced are today in efficacy studies in animals. And if you go up the mountain of the funnel, we have more than 50 programs that have ongoing chemistry to bind to the right target and that are selective to other targets.
Mm-hmm. And the bet being that you can go through all these steps much quicker than using a traditional drug discovery approach, because you have some guarantee about the selectivity and the therapeutic efficacy of the compound from the start.
No.
Okay.
No, that's a very important point. And in fact, I don't think that being faster by 6 months or even 1 year or cheaper by a few millions in the discovery phase, I don't think it makes a difference.
Okay.
What makes the difference, in my opinion, is the ability to invent molecules that are truly innovative for targets that are not well understood, for which there is no candidate. So new targets, new programs, hard drug targets, etc. So, and in fact, it leverages something very important at the heart of Aqemia, the fact that we don't use AI. So, but physics. Let me explain, please.
Yeah, go ahead.
If you work with AI, you need to train on solutions that already exist. So what you do is you typically take a program in oncology that is already known, this kinase everybody knows about, for which there is a lot of data all around, molecules that have been designed by medicinal chemists in the pharma, published, patented, etc. All those molecules that are public. Then you train your algorithms on this data, and then everything you can do is fast followers, me toos, molecules that look like what you trained on. That's absolutely not what we do. We tackle problems for which there is no data to train on, no known solutions, no known molecules. And for them, we craft molecules that nobody had ever seen or invented, and so we build, we are the only ones, I like to think, the only ones being able to invent the right molecules for those problems that have no solution today. And thanks to quantum and statistical mechanics. So physics.
Understood. So it's really...
So sorry, to summarize, because it was a very long answer. The key, in my opinion, to building something very useful to the world and to inventing a new biotech, a new TechBio, is not to be a bit faster or a bit cheaper because of AI gain of efficiency in inventing the same molecules as were already existing. The key is to invent those molecules for hard problems, new problems, and with, we hope, better chances of success thanks to highly selective and highly efficient molecules. And I think the physics that we have is the key discriminant to building that.
I think it's a very interesting take, because what I've heard so far is, improvements in terms of speed, improvements in terms of cost, but no it was not like saying, let's tackle like those, let's say, unmet medical needs for which there are no compounds yet or no solution at all, and bring a solution to it, not regardless of how much it costs and how much it takes, but increasing the chance of success of, providing a new therapy for that. So, and I think it also explains how your approach is different than what other, actors are doing. Because as you said, like you do not build on, leveraging the molecular structure of millions of compounds existing to build a model. Like you just start from purely chemical and atomic level interactions to create those new compounds that are not, let's say, biased by, a model that just that is based on existing compounds.
Yeah, you say it perfectly. In fact, we go beyond the limits of artificial intelligence, which is to only train and extrapolate, interpolate between the training points. Yeah. Maybe I can give you a very short example, coming back to those CDK2 and CDK4.
Yeah.
say, and that's not absolutely wrong. It's a very good example. Today, most of CDK2 there are a lot of molecules that bind to both CDK2 and CDK4. So the literature, the scientific literature, the patents, the world, the internet is full of molecules that bind to the two targets, CDK2, CDK4. Say no that you want to invent for a very good biological reason, and that's the case, in fact, today, a CDK2 selective molecule. You want a molecule that binds to the CDK2 but not to CDK2. Your AI black box, if you trained on binders of CDK2 and 4, will only invent molecules that bind to CDK2 and 4. It doesn't know the difference between CDK2 and 4. For us, CDK2 and CDK4 are completely different stacks of atoms.
Yeah.
We invent the right molecules that will do this interaction with CDK4 and not this interaction with CDK2. And so it will be selective. So in fact, once again, we go beyond the limits of AI that needs to train on data, so on chemical matters that already exist. I like to think that some others may invent molecules that look alike, are me-toos for fast follower programs, while we invent completely new chemical matter for truly innovative programs.
Understood. And so for those internal programs that we talked about, at which point do you, because obviously you're at this discovery phase of the process, and at some point, you go through these preclinical trials and then you go into clinical trials. Is that something that you intend as a company to, do you intend to manage the whole end-to-end process? Or would you at some point partner to run those clinical trials based on the compounds that made it to this stage?
So yes, we want to be part of the full process up to validation of phase 2. So validation that the molecule is useful to a patient.
Yeah.
Then the way forward, especially for the first programs, I think we are not there yet, but it's very, very soon. It will be for the end of this year or the next year. Maybe the right partner with the right knowledge to fast forward what we would have done, learning clinical trials, is the best solution. It's maybe we will look for a partner that will increase the chances of success of those trials because they know they have already done it in the past for similar indications or something like that.
Okay, and I guess is your strategy to demonstrate, because you mentioned, initially that your technology is applicable to any conditions, like any disease areas. It's not specific to oncology or neurology or something different. And so I'm wondering, how do you balance between, pushing, these ongoing programs that you have and also accepting potentially requests for other indications that you're not focusing on right now? How do you make that balance?
So there are several balances to find. The first one is between the internal portfolio and our portfolio with partners. We have ongoing research collaborations with pharma companies, and we typically limit ourselves to 20% of the team, no more than 20% of the team working on programs in partnerships with pharma companies. Those partnerships are extraordinarily good to learn, validate the technologies to the outside world, and to do revenues.
Yeah, for sure.
And so 20% of the team is working on those programs. And then for the rest of the team, the fact that we have this mathematical solution, the physics that we do that is unique thanks to the mathematics at the origin of Aqemia, allow us not to, we don't need experimental data to start with, but the structure of the therapeutic target. And we don't need supercomputers to do brute force computing with GPUs, etc., as it is needed for AI first companies. So we are very lean and able to start many programs in parallel. So we are a portfolio company. And then there is this risk-reward balance. There is this, for the wholly owned pipeline, risk-reward, therapeutic areas. So mostly it's 80% oncology today, 20% CNS and metabolism. And does it answer your question?
Yeah, I think it answers my question. So I think the trade-off, 20% versus 20% working with external companies, 80% internally. I think that's, it's interesting because it's still for the potential of the technology and also even managing the partnership. I guess the one with Sanofi is one of these partnerships that is part of this 20%. I would say having, like you said, you were 70, so 14 people working on these partnerships. To me, it sounds like it's probably a lot of work to manage for a small amount of people. And I think it's interesting to hear how you guys are distributing these efforts in the current team. But I assume that going forward, you guys are, and I saw you mentioned you recently expanded to the UK. I guess that comes as well with, opportunities to, broaden the size of the team with some of the talent there.
Exactly. We are quite well funded. We raised in the past 5 years 100 millions and 2 times in the past year. And the last time we raised was especially to increase the size of the team, to be able to tackle more programs in parallel because the technology can, and the deal with Sanofi especially, it's a large deal, was absorbed in terms of how you manage things. So at the beginning of this year we opened the London offices. It's now 12 to 15 people. And we are 70 people in total, and we want to be around 90 at the end of the year and around 150 at the end of next year.
Okay.
So it's indeed a lot of bandwidth a challenge to attract the best talents everywhere in Europe and have them in London or in our London or Paris offices.
And can you tell us more about, the types of, let's say, roles in the organization for now? Because I'm wondering, because that's something we talked about with Jean-Philippe, and he was saying it's really hard to find people with, it's like 2 problems. Like one was saying it's hard to find people who have a very strong biology background, but have also the computer science competencies. And the other way around is also a problem, saying we cannot easily find people with very strong computer science skills that are already, let's say, familiar with biological processes.
So I share this observation, but we have a strategy around that. First, we look for T-shaped people. So people with strong expertise, be it in biology, chemistry, mathematics, computer science, machine learning, or whatever, and a capacity to connect to the other expertise. And we have internally, I don't know how to say that correctly, but people development, efforts so that people internally have the time and the resources to learn the vocabulary of the other expertise. So our strategy is find people that have their expertise, are able to connect, and give them the means, the resources to learn the part of the other expertise that is needed for the job, and make it fun, pleasant to connect with those colleagues of you with another expertise. Does it mean something?
Yeah, makes sense. Maybe it inspires Jean-Philippe, and he can copy that approach.
Let's see. He's a friend I wouldn't dare give advices to.
My other question was, how far are we from, seeing one of the Aqemia drug candidates that are part of your, your own programs? I mean, first go into human clinical trials and, potentially looking even more in the future, like being available, in the market?
This is a very difficult question. And the why it is so difficult is because we must stay very humble with respect to biology, with respect to the human body, with respect to the unknown, and that's also the beauty of science and the biology, etc. So I can give you my what I would love.
Yeah,
but
your wish.
My wish. And that's really the core of the why we are here. So my wish would be that we enter into clinical trials next year with a molecule that we designed from zero to a molecule that binds to its target, doesn't bind to the others, have good, we say ADME, so physical chemical properties, and shows efficiency in the cells, and then in the animal models, and then show it in a regulatory phase, etc. So I wish we enter into clinical trials end of next year. It would be absolutely great. And then when it would be available to patients all around the world, this I would love to be by 2030.
2030.
Yeah, in fact, I like to think of Aqemia like several phases. 20 to 25, build the platform, build the technology, start from fundamental science and mathematics, and build something that is useful to, being confident we can be useful to patients. Build the tech and build the drug discovery processes, etc. Then there is a second phase, which is 26 to 28. Learn clinical development. That's another really...
big piece.
Yeah, that's a big piece. And then I hope repeat. So build a platform, know how to do drug discovery very well in useful programs, and then put them into patients, test, be useful to something again, and learn, and then again and again and again.
For a lot of different indications. No, that's that's everything I wish you as well. Maximilien, I'm very thankful for, your time so far. I'm keeping an eye as well on how much time we have left. One of the first, recurring questions I had for, every guest on the podcast is, for those, of which we sparked interest for, the field in which you work, which is very, specific and I think like interesting. What resources would you recommend, them to have a look at or to explore, to learn more, especially if they don't know much about it? So, are there any books, publications, or websites you would like to redirect them to?
So if they want to learn about Aqemia's technology, for instance, that's what
exactly. Or yeah.
So I would suggest some papers we wrote in my group in the Journal of Chemical Physics. This is theoretical pieces, but or in physical reviews. We have more than 47 papers, peer-reviewed papers on the theory, etc. That's the hardcore science.
Yeah.
Hardcore. And then I have a very naive answer. ChatGPT is extraordinary.
Really?
Really. Much better than I am at communicating science. Since it is public, it's extraordinary.
Okay. Yeah. Then I'm happy to link some of the papers in the show notes and also link ChatGPT. The second question is, and it's a bit early for you to answer, and maybe you will have the answer in 2030 if your wish is fulfilled. But can you share with us an anecdote from your work so far with Aqemia that question is normally like that made you realize the impact you were having on people's lives, but in that case maybe it's probably the impact that you've had on industrial partners or what made you? Yeah, do you have any anecdote to share in that regard?
I think it's a very easy one. The first time, really, the first time we had a positive result from one molecule we designed that reduced the size of a tumor in a mouse. Can you imagine for the team and I and my co-founder, Emmanuelle Martiano, we had started from pure quantum physics and mathematical stuff. And one day you receive an email with a figure showing that your molecule decreases the size of a tumor crafted on a mouse. And then you start to imagine that it will maybe do the same in patients, etc., etc. So it starts to becoming true. I don't know if it makes sense, but
no, I think
I don't know. We were like 50 people in Aqemia at this stage, or 40 people, and everybody was quite wow.
Yeah.
Wow.
Touched.
Yes.
Yeah.
It was. We were quite crazy. Like
this is working.
And at the same time, very humbled with everything we have to do. But it was a huge milestone.
Yeah. Thanks for sharing that. So if you would, recommend a fellow, innovator in healthcare, in health tech as a potential guest for the podcast, who would you recommend and why would you recommend her or him?
I would recommend Fanny Jolin. She is the CEO and co-founder of Oracle Oncology.
Okay.
And she works at Institut Gustave
Roussy,
specialized in cancer. Okay. And she's an expert in molecular oncology and in organoids.
Okay.
And for me, I come from StatMex, so theoretical stuff. I've seen tumors from patients, in those plates and test molecules on those real tumors. It was yes. So and Fanny is an extraordinary researcher and an example CEO for me.
Super cool. No, I'll definitely consider it. I think it was like episode 13. I talked with was it the chief scientific officer of Mimetas? This company. Yeah, it's a Dutch company that is very strong in, organoids development that I think got acquired by Merck. I also know that I work I work for Roche, and I know that in the drug discovery process, we push also very hard on the use of, organoids, and I'm like fully with you. It's a very interesting technology that they're using. Yeah.
In fact, yes, imagining a path where you can be absolutely sure that a true tumor is reduced by your molecule before going to the patients, and the beauty of its complexity. And I think it will completely change the drug discovery process. Really. The chances of success, I hope and I believe the chances of success, the probabilities of success will be dramatically changed by even a few %.
Yeah, but still a big change. Yeah.
Fanny, and she's very cool.
Awesome. Then, thank you so much, Maximilien. I learned a lot of things, and I was really happy we could dive into all of this, because for now, there isn't a lot of information yet on the website, or exactly. So thank you for your openness and sharing everything you've shared today. You told me it was the first podcast episode you've done, so I hope it was a positive experience for you. But it was a great conversation for me, so thank you.
Thank you very much, Mathieu.
Thanks for watching the episode. I hope you enjoyed it. You can subscribe by clicking on the podcast channel at the top right and watch another episode here at the bottom.